Hello everyone, my name is Merri-Grace and my presentation is on the evolution of the major histocompatibility complex. Every day in our bodies there is an invisible battle being waged. Thousands of microbial pathogens fight to penetrate our bodies and lead to infection, and our bodies have evolved sophisticated means of preventing this. We have physical barriers such as skin and mucous membranes that trap and block pathogens. We have cells in our innate immune system, such as neutrophils and macrophages that engage in phagocytosis and consume pathogens. And we have the adaptive immune system that has evolved a sophisticated means of identifying specific pathogens, remembering them, and leading to customized ways of attacking them. And this is done primarily through the major histocompatibility complex. So what is the major histocompatibility complex? The major histocompatibility complex is a region of genes that code for proteins that present antigen peptides to B and T cells of the adaptive immune system. This leads to disease resistance and immune memory. This presentation will explore the role of the MHC, its origin, the selective pressures it faces, and how it has evolved to play a role in autoimmune disorders. So what is the structure and function of the MHC? It is hundreds of genes that are located on chromosome 6. They're organized into multi-gene families and that are categorized into 3 classes. Class 1 codes for surface proteins on almost all cells that present peptides of intracellular pathogens to cytotoxic T cells. When the cytotoxic T cells recognize these peptides, they destroy the cells that have been infected by intracellular pathogens. In class 2, only certain immune cells have these surface proteins, and they such as dendritic cells, and they present to certain cells of the immune system, such as B cells, and that leads to activation of the adaptive immune system and the production of antibodies. And then in class 3, these genes code for proteins involved in other immunity functions, such as the inflammation pathway. Not all functions of the genes in the MHC complex have been identified, but there's ongoing research in this area. So the origin of the MHC. Scientists believe that a Big Bang event occurred and that there was several large-scale duplication events that led to the creation of the MHC complex. And one piece of evidence that supports this is pyrologous regions. Okay, so there are 3 chromosome regions in humans that are very similar to the MHC. On chromosomes 1 and 9, they contain segments of DNA that are pyrologous to the class 2 and class 3 regions on chromosome 6. And there are several specific examples of genes that are found on chromosome 6 and are similar to genes in other areas of the genome, such as RXR receptor family, Notch homolog genes, and PBX genes. So anchor genes are further evidence of these large-scale duplication events. As I said before, genes of the MHC are organized into multi-gene families, and anchor genes are located right next to these multi-gene families. Anchor genes are regulators of gene expression and duplication. They evolve at a much slower rate than the other genes of the MHC. Another interesting facet of these anchor genes is that they're found in non-vertebrate species, while the MHC are only found in vertebrates. This can be interpreted to mean that these anchor genes are a remnant from before the duplication events. So in jawed vertebrates, class 1 and class 2 regions have been found. The oldest member of this group is cartilaginous fish, such as sharks, and it's interesting to note that all defining genes of the MHC, such as T cell receptors, immunoglobulins, MHC peptide proteins, are found in the oldest of these jawed vertebrates. Cyclostomes, such as hagfish and lampreys, these are jawless vertebrates, and they don't have any of the defining genes of the MHC. Thus, we can conclude that duplication events that resulted in the MHC must have occurred around the same time that jaws developed, as you can see in this picture. So What are the selective pressures on the MHC? MHC region is highly polymorphic, and one of the most common forms of polymorphism that are found there are single nucleotide polymorphisms. These are single base pair replacements. And balancing selections that increase the rate of these polymorphisms are sexual selection and pathogen selection. So they found that sexual attraction and mate selection is partially MHC dependent. In particular, in mice studies by Pens and Potts, they found that through olfaction, mice were most attracted to mates that had MHC complexes dissimilar to their own, and they believe that this is an evolutionary mechanism that prevents inbreeding. Also, in humans, they found that high levels of MHC similarity between mates can increase the risk of spontaneous recurrent miscarriage. Another type of balancing selection that contributes to the high polymorphism of the MHC is pathogen selection, and this is explained by the overdominance model. The overdominance model postulates that heterozygous individuals are better able to recognize and respond to a variety of pathogens compared to homozygous individuals. One study showed that MHC heterozygous offspring experienced increased fitness when they were exposed to various pathogens, and this goes back to the Red Queen hypothesis, which states that as pathogens can rapidly mutate, there's an evolutionary arms race, and genetic MHC diversity of the host increases and mutates, and this increases the chance that they will have a rare allele that will protect them from the pathogens. However, in populations, pathogen selection is not always consistent across different generations, and when pathogen selection's minimal, genetic drift tends to be homogenizing, and it reduces the diversity of the MHC complex. This is especially seen in small populations. So how does the MHC complex relate to autoimmune disorders? So they have found that many of the susceptibility loci of disorders such as diabetes, multiple sclerosis, lupus, myasthenia gravis, rheumatoid arthritis, and hundreds of others have been attributed to genes found in the MHC complex. Now, autoimmune disorders can be defined as any disorder that leads to the loss of immune function and the breakdown of self-recognition. This leads to chronic inflammation, autoantibodies, and the loss of tolerance to self. And it has been very difficult for scientists to attribute specific mutations in the MHC to specific autoimmune disorders. And this is attributed to the highly polymorphic nature of the MHC complex. In fact, the HLA-B gene of the class 1 region is the most polymorphic gene known to man. And one of the theories that they use to explain this autointolerance results, they believe it's from mutated class 2 peptide presentations of either foreign antigen or self to self-reactive T cells, resulting in apoptosis and inflammation. And their lack of understanding in this area makes it really difficult to treat autoimmune disorders. Now, it's sort of an interesting paradox because even though autoimmune disorders usually reduce fertility and limit fitness, the incidence is actually rising worldwide. So several scientists have suggested that perhaps there is some benefit to the risk alleles of autoimmune disorders. A recent study of Roma genetics, which are a gypsy population, supports this hypothesis. In the 14th century, the Black Plague stormed through Europe, killing millions of people. An immunologist named Mihail Nitia and his colleagues studied the genetics of the Roma people. This group's ancestors migrated to Europe from India before the Black Plague occurred. They compared their genomes to individuals that stayed in India, and they discovered that there were changes in 20 genes that the European Roma had, but the Indians didn't. 3 of these genes coded for toll-like receptors and played a role in identifying and eliminating dangerous bacteria. However, some of these genes were also involved in inflammation and autoimmune disorders. And this research might explain why there's a higher rate of autoimmune disorders among Europeans compared to other ethnicities. And then when analyzing loci that are associated with other autoimmune disorders, there's evidence that selection has been driven by the coevolution of pathogens and hosts. For example, FCGR2B gene found in the lupus susceptibility loci plays a role in regulating the immune reaction to a plasmodium that causes malaria. And then another one is the P2PN22 non-synonymous mutation. This leads to a variety of autoimmune disorders, and in this case, it helps protect against Vibrio cholerae, and this pathogen has led to the conservation of this mutation. So, in conclusion, understanding the MHC complex shapes the way we understand how our bodies defend us from infections and how it sometimes fails us. As scientists continue to untangle the mysteries of the MHC complex and how it evolves, they will develop a clearer understanding of the mechanisms and pathogenesis of autoimmune disorders. As these mechanisms are elucidated through research, new treatments can be developed to aid our immune system in its invisible battle. And to conclude, this review has explored the role of the MHC, its origin, the unique selective pressures it faces, and its connection to autoimmune disorders. Thank you for watching. Have a Merry Christmas.